Okay, and we're recording. So good morning, everyone. Happy Monday. Welcome to week 2 of the protein crystallography collaboration. This week, we're going to meet Monday, Wednesday, and Friday, the same time, same place. For today, we're actually going to do two things. Mrs. Mackey is going to explain a little bit about what lysozyme is, what it is, and why we are going to be looking at it. And we're also going to take a review of the PDB scavenger hunt. So I'm going to let Ms. Mackey take over and start up. So good morning.
Okay.
Right.
Good morning, everybody. Everybody can hear me. So Mr. Boland, since we haven't done any practicing with this at all, I'm gonna do the present now, and you're gonna tell me what to do to get the highlighter
little pointer thing. Okay.
Right. I'm in sharing the screen.
Okay.
And do I select a tab? Is that what I should do?
Yes, select the tab that you want to share. Yep.
Okay, and we wanted to start. You wanted to start with lysozyme, you said.
Yeah, we could do that. Yeah.
Okay.
Sure. All right, now present your slideshow like you usually do.
Present new slideshow, like just.
Oh, so go to the tab that the slideshow is in, and just click slideshow. So this way you can get the full. Yeah, there you go.
Okay.
Now go back to the Google Meet tab. So you should see, right? And then on the bottom, do you see the little
the arrows?
Yeah, there's arrows and there's like a little thing that you can use to you can click on that. Yeah.
Okay. And you just do that and then it just goes away.
Yep.
All right. So I'm gonna go disappearing ink.
Yep.
And let's hope that works. All right. So good. Sorry guys, I'm learning on the job. But luckily I have absolutely no pride, so I'm not embarrassed at all. I don't know.
It's summer, we're all good.
Yep. All right. So lysozyme is up now. No thanks. Oh, wait, try scrolling and zooming. I'm not sure what that's doing. Let's see. Allow this.
Yeah, you can just hit no thanks for that. Yeah.
Oh, too late. I did it. I just did it. All right. So lysozyme was the right click to change the tool. Oh, there's too much going on. The lysozyme was the first enzyme to have its structure resolved. It is, see, now I'm annotating. Okay. Roll it. Go. Okay, that's what I said. It is an enzyme commonly found in animals, and it serves as a defense against bacteria. So a lysozyme is a catch-all word. To lyse means to split, and zyme is the suffix that you would put at the end of any kind of an enzyme. So that's what it does. It basically breaks up different things, and I'm not sure if it's the cells of the protein. I think the cells of the bacteria. And it provides a defense mechanism. So it's found in human tears. Our immune cells make it. Just about any animal makes it. And it was first isolated and resolved by David Chilton Phillips. And he used chicken egg whites as his source. And he resolved the structure in 1965. I'll be perfectly honest. I did not have access to the paper. I tried getting access to the paper, but they were going to charge me for it, and I didn't have the time to get the whole thing. But he deposited it in the PDB in 1975. Essentially, what he did was he began the field of protein crystallography. X-ray crystallography had been used for a really long time. Everybody who's familiar with the story of the discovery of DNA knows that Rosalind Franklin used X-ray crystallography to get a picture or an idea of DNA's helical structure. But the protein, the first enzyme to ever be deposited, the first protein was not this. It was something different. Was resolved in, like I said, 1965, but the PDB didn't exist because the internet didn't exist. Honestly, I'm not even sure in 1975 when Dr. Phillips deposited this, what he deposited it to, because the internet didn't exist in 75. But he did deposit this structure in 1975, and it's got, if you've been with us for a long time, you know that all of the proteins in the PDB have a 4-digit ID number, and this one starts with the number 1, so it's got to be super duper early. That being said, the most recent deposit of lysozyme happened last week. So its structure is 8km. This is the 1975 structure here. And this is the one that was deposited last week. So clearly, it's still an important protein that people are doing lots and lots of work with. So what we're going to be doing is using lysozyme for you to learn the process of growing protein crystals. At your schools, you will be growing the lysozyme crystals under a bunch of different conditions. And this will allow you to learn to do things that you may or may not have had a chance to do in your classes, like work with micropipettes. I find that depending on what grade you're in and what sciences you've taken, you may or may not have worked with micropipettes, and it's an important skill that you're going to need. How to actually create the plates. Depending on the equipment your school has, you may do hanging drops, you may do sitting drops, you may do both. You also are going to have to learn to keep track of metadata and get familiar with all the different metadata that there is out there to be collected. After a certain amount of time, you will then look at your plates and screen them to see which conditions actually have crystals. So here's an example of what you would be doing. That's about all you can get done at your schools. Then you can bring the plates with you once you've been badged. And at the end of the week, Dr. Pepinella is going to go through the very long and complex process of getting badged. It's not difficult. It is just arduous. There's just an awful lot of steps. But once you're badged, you'll be able to go on site to Brookhaven National Lab, and there you can use their equipment to fish for crystals. You might actually be able to fish for crystals. I had my kids do this in our classroom too, but there was nowhere to put them because we don't have liquid nitrogen. So the real point of fishing for the crystals is to then preserve them, get them in those loops, preserve them in liquid nitrogen, and then you have them stored in those pucks to be taken to the lab. On a different date, when we have beam time, what we'll be doing is using lysozyme as the starting point for training on that day's beam line. So the first thing we'll ask that robot to load into the X-ray beam will be a lysozyme crystal. And this is how you will be taught while you're on site to quote unquote drive the machine. And that means to operate all of the controls that are at that particular beam line. We have FMX and AMX and NYX. And there, as Mr. Boland was saying last week, they get updated there. They get new software, they get new equipment. So just about every single time we go, there's something new going on. So we figured lysozyme, we know what a crystal looks like. We know how to do it. It'll serve as everybody's training session when we are actually on site. It'll also, and this is probably the most important thing, show you what a good crystal looks like under when the X-rays actually go through it, because we have spent a whole day, 6 hours shooting our experimental crystals and not actually seeing any good ones. And for a student who doesn't have that many beam times in them, it's kind of sad. So here is, we did this in June and it worked really, really well. Here is a person shooting a lysozyme crystal and getting a really good look at what a nice formation looks like. And then we will still collect that data, because sooner or later, you're going to have to know how to take data from the beam and put it through Qt and Phoenix and refine a structure. And what better way to learn how to do that than using something that we already know exactly what it's supposed to look like. So we'll then use lysozyme to do that. And that's basically your workflow as far as the starting point of everything. We're going to use lysozyme so that you know how it all gets done. But by now, those of you who've been with us know that you're not really growing the crystals. That we have Dr. Lovell at Kansas University, and he's growing the crystals that you're going to be working with. But thanks to Rockmaker, you can screen the plates. Just like it will be a remote experience, but once you've actually done them in real life, it'll obviously make a lot more sense to you. Not that this doesn't make sense, but it'll be a lot easier to relate to. And then using Rockmaker, you can, are we going to do this? Are we going to post the link to our classroom?
Yes.
Okay.
Yep.
So I'll leave this, and when we post the link, I'm sure we'll add this. There is a standard login. It's lowercase Spark. I'm going to point out that the password, this thing right here, is a lowercase L. It's not a number 1. It just always messed me up. And so this year we are focusing on 3 proteins: S11, S12, and S13. And I did not know how deeply we wanted to go into these because each school is going to be focusing on one. So since we haven't divvied them up completely yet, and I haven't had the time to look into them, other than their names, I don't know a whole lot about them. I did go over them briefly last week where we talked about how S11 and S12 have relatives that are already deposited in the PDB. So that's kind of a starting point for some people. But S13 looks like a really unique protein that we're going to need to do a little bit more digging on.
Yeah, we don't need to go that deep into them. That's the reason why we're going to do our lit searches. Yep.
Yeah. All right. Okay. So then once we've established which crystals it is you want, Dr. Lovell's lab will be the ones in charge of fishing them, cryoprepping them, putting them in a puck, and then the pucks will be shipped to us. And then once at the beam, you'll be able to do everything that you did with lysozyme, only now with real proteins that are experimental, and hopefully use that data to resolve the structure and then deposit into the PDB. So that's the idea, is take everything that you learned working with lysozyme and apply it to a novel protein now. And I think that's it for my lysozyme presentation. I don't know if you were looking for more from me, Mr. Bolin, but that's what I got.
Perfect. That's it. And again, that's what we want is the students to understand that the lysozyme, although we use it as an exemplar, it is still an actively researched protein. And this might, who knows, if we get a really good, sometimes we do this just, we're training to see how this looks. If we get a really good crystal and we get some really good diffraction, that could be something we deposit into the PDB for whatever reason. The other thing too is we can start looking at, and I think you mentioned this, one of your students might be looking into ligands.
Ligands.
So, and that's a really nice study to do. But, it's amazing how this protein is still being actively looked at since 1965, and they're still going at it, and they're still working at it. So it just goes to show everyone that, just because we finish work on a protein doesn't mean that the research is done. That's actually just the beginning. That's the door opening to allowing others to start corroborating and doing the same work, and then also taking our work to the next level and doing different types of research on it. One of the things we could do, which we haven't mentioned, and I don't think this is something we can do, but what scientists do is they mutate the protein very often. They'll change 1 or 2 amino acids and then shoot it and look at the structure and see how did that mutation affect the structure. And the reason they do that is partly to determine what areas of the protein might be essential to the structure of the active site or the binding site for a ligand. And so if they can knock out 1 or 2 of these residues and alter them, that could potentially result in altering the structure and the function of the protein. So a lot of times those are interesting things to do too, but I honestly don't even know how to go about doing that, other than maybe you take your plasmid and you mutate one for the base pairs, and then that generates a mutated protein. But that's something that's what scientists can do with the work that we do.
Yeah, I honestly don't know how they purposely mutate either. However, one of the things when we go into the next presentation, which is about the protein data bank, looking at lysozyme in the protein data bank, there are over 5,600 entries, and most of them aren't human. There's only maybe 7 or 8 that are human. Wow. And the rest are if you're isolated from it, or if the gene that creates it comes from a different organism, there is going to be an amino acid difference here or there. And comparing them could also be, I suppose, a study, and whether they bind with ligands and a whole bunch of other things. So, yeah, there's a lot.
Right. Thank you.
Okay.
All right.
So now to change presentations, unless anybody has any questions about this.
There she is, the graduate. Wanted to say hi.
Yeah, I did this program like 2 years ago, something like that. I did this program 2 years ago, so it's very good. You guys have fun.
All right. All right, you can switch it.
Okay.
Go ahead.
Okay, switching. I'm going to stop sharing and then switch to the other presentation.
Okay. Did anyone have any questions while we're switching? Or a comment or something to add? No. Okay. All right. I see that.
Okay. Yeah, and I've already forgotten how to. Oh, never mind. Got it.
Got it. Okay.
Good. We're good. Okay. So for as of this morning, well, as of the last time I looked at it, we had about 9 answers to the PDB scavenger hunt. So some people didn't do it, and that's okay. It's summer. 2 of them were teachers. So, and as we're going to see through this, it is a complex website. It's a rich, deep, rigorous website with so many different things. And there may be more than one way to get an answer. And I'm going to be honest that I'm not an expert in the PDB website. So I got my answers, and I know that I got right answers because, well, while there may be more than one way to get a right answer, there really should only be one right answer. You may get to it multiple ways, but it's interesting that a lot of our questions did not have 100% people getting all the answers right. So it's important to kind of go through where things are. I find the site kind of intuitive if you read carefully, but a lot of people didn't. So we're going to go through the questions one at a time. The first question was to do a basic search for TRMD, and it asked how many structures were published from this specific bacterium. And the answer is 3. So if you put it up in the search bar, you put in TRMD, and over on the side of the site, you have all of these different things. You can scroll down and make it as much as you can. Here you had this one, which is a certain strain, and then just the plain Anaplasma phagocytophilium. They're there. So the 3 structures, what's the latest to be published? 2 of them were from Seattle, and they were older. And the most recent one was from us, which shows up as 8SH4. And just about everybody got this question right. And then the next question, unfortunately, talked about structure 6UEV. And I had not checked. I did not know that this had happened. The 2 questions about it that we're starting off with, what resolution was it resolved in, and what organism was it produced from? Before we deal with that, we have to deal with the fact that it was a trick question, that 6UEV has actually been replaced by 8SH4. So, and it's just, you don't have a choice. If you plugged 6UEV into the search bar, it automatically brought you to 8SH4. You didn't have a choice. There was nothing else you could absolutely. Yes, Mr. Bolin.
I was just going to say the reasoning behind that was it's the same structure. I found out that the group that was working on it is from our group. There they are. And what they did is they had to make some revisions to this, and it had to be resubmitted. And because they had made some substantial changes to it, they had to give it a different number. So this basically supersedes it, but it's the same structure. The resolution is the same. There were just a couple of things I believe that they had to adjust in the process. So that's the reason why.
You explained that to me, and I'll be perfectly honest that I'm a little confused because while if you scrolled all the way down, so you type in 6UEV and you get to this page, you scroll all the way down, and this circle tells you, right, the text in here tells you that it replaces it, but the version is still, there's only one version. Right. So why doesn't it say that there were 2 versions, 6UEV and now this one? Or is it because the version would be a purely different protein?
So what happened is this is the version of this protein now. They gave it a new PDB ID number. So 6UEV has been eliminated. So it's just in there in case people were looking at that because it may have been, like we had posters with it and everything.
Right. Like the old exits on the throughway. Right,
exactly. Right. They'll still put that up there so people know what it is. But this is when it says version, this is the first version of this one. And so 6UEV no longer exists as a separate structure. This is what 6UEV has become. So, and you can see
Okay.
they did it on my birthday. Oh, how about that? Look at that. So I went back through and I found emails on this from the PDB that they had sent me emails to tell me like, oh, we're making this change. And it just never clicked that, oh yeah, we need to adjust it.
Okay, so now we will. Absolutely we will. Okay. So everything we're going to be going on now with, you're done, right?
Yeah, I'm done. I'm hitting the space bar. Sorry.
is what you answered for 6UEV is going to actually be then 8SH4. It's been resolved to 2.9 angstrom, so we know there's work to be done there because after last week we know that we're aiming for 2 or lower, but that's the way it was. It was done by X-ray diffraction, and it was produced in E. coli. So looking at people's answers when sometimes people got things wrong, I think maybe we need to just go over the difference here. So the organism that normally produces the protein in nature, the one that you'll find the gene for this TRMD, is the Anaplasma phagocytophilium. But rather than getting the protein directly from that bacteria, we put scientists put the gene into an E. coli through genetic engineering and ask the E. coli then to make the pure protein for them in the lab. So when they talk about the expression system, that's the difference there. It's not a naturally occurring protein in E. coli, but that was the bacteria that was used to make the purified protein. Another question that came up with it is what ligand is also present. And if you scrolled all the way down, glycerol is the ligand that they talk about, and that's right there. And then at what temperature and pH were the crystals formed at? Most people got it. One person, I think, didn't. And in order to do that, the temperature was 298 degrees Celsius and the pH was 4. You have your experimental data validation, which is right underneath all the main stuff at the top of the page. But you had to scroll all the way down. And at the very bottom, you had to view, because if you look at all of this, temperature isn't here, pH isn't here. You had to click this, and it brought you to more details. And then the more details involved the pH and the temperature. So now we move away from the protein that we deposited, and now we're just doing a basic search for insulin. And the directions were to use the polymer entity description in the drop-down menu. And I think at least a couple of people got a little lost there because they didn't get the right answers here. So if you go to the PDB and start typing in insulin, this drop-down menu automatically shows up, and it's huge. So I had to scroll down to get to the polymer entity description thing right there. And when you click on that, I clicked on just the plain insulin and got 166 entries. Of them, 113 were from humans and 13 were from pigs. And I'm bringing it up because you were asked about pigs later on. And at least one person said they had to look up what the Latin name for pig was. So there, for those of you who didn't know it, it was Scrofa. So that's the answer so far for that. And yes, and this is how you find exactly how many of them there were. So if overall there were 166, and then it breaks it down over here by species, 33, not 13, 133. Yeah, that's a typo. It's supposed to be that's 333 from humans, and then 13 from pigs. And they're the pigs. And then various ones from other organisms. The newest human insulin submission. So the idea behind this question was to get you to find the filter for how to find the latest deposit. And the same thing for, and then locate the resolution later. So whenever you're doing that, the most recent is right here. So you put in the insulin in your your search engine, you go into humans over here, you click on this little thing, You click the human Homo sapiens, and then you click this thing, and it brings you to to a general page. But then you want the release date, newest to oldest. This drop-down menu gives you a bunch of different things you can look at. So by doing that, your newest one, your most recent one just pops up, and it's 8 RRP, and it was resolved to 2 angstroms, and it was done on July 10th of 2024. So fairly recently. So like lysozyme, you would think insulin is something that's already a settled science, but apparently not. The 4-character ID for the highest resolution of pig insulin, same thing. So now you have to go and click the Suscrofa over here and then run your search. And again, going over to this area over here, you change you select from the drop-down menu, instead of newest to oldest, the best to worst, and the best resolution pops up. And that's how we found out that resolution is 1.46 angstroms. I think I have it there, but I covered it with my graphic. Then looking for protein 4 INS, what organism was it from and when was the structure published? It was isolated from pigs on April 15th of 1990. Almost everybody got that. I don't know the one person who got it wrong. I don't know how you got it wrong, because this was basically just plugging 4 INS in and getting it, unless you did the lowercase l. Or maybe that is the lowercase l and not the uppercase I. I'm not sure. How many amino acids are in the molecule? And here's where I'm going to say I'm not sure what the right answer is. Here's what I found, and I'm going to have Mr. Boland help me out here, maybe Dr. Pepinella. The first answer I came up with was 102, because when you look at the basic structure here, down here at the bottom on the underneath side of the 3-dimensional picture that you have, one of the things that you have that pops up is, I hope you all can see this. I have a whole bunch of stuff in my way right now, but it says number of residues 102. It says, and I'm like, oh, okay, that sounds right, except if I go to the actual information on over here in this area, and I look at that a little bit closer, I get 21 residues in chain A and 30 in chain B, and that does not add up to 102, so I am confused. When I look closer at the chain A and the chain B, that's all there is. Here's chain A with a total of 21, and I'm clicking on it, and I went through, as you click every single one of these. Here I've got one of them highlighted. It highlighted it in the model for me. I clicked all the way through it. Nope, there's only 21 there. And then I go to chain B and the same exact thing. So I'm not exactly sure.
I think it's a dimer. Some of these insulin molecules will form dimers and trimers.
Okay, so 51 and 51
why.
is 102.
Right. I think that's the reason why. Yep. But they're only showing in the PDB, they're only showing the primary chain. But I think this one forms a dimer. And there was another one, I remember years ago we were looking at it, and they actually had one that they did something to it with a ligand, and that actually made a trimer, and you actually had 3. And the whole idea about that was that doing 3 of them actually slows down the insulin's activity. Because I think these studies were done with trying to modify it so that when people were taking their insulin shots, they weren't getting a sudden rapid drop in their blood sugar, but that it was causing this, it was basically reducing the efficiency of the insulin in allowing the glucose to enter the cells. I think that's why they did that. But I think this was a dimer. I think that's the
so.
Yeah.
Legitimate question from the person who doesn't know. Did I miss somewhere in the big overall information that they give us about this protein that they tell you it's a dimer? Was it there and I just missed it?
I have to look at it because it said something about heterotumer, hetero2-mer. So I'm wondering if there was like a shorthand like heterodimer. I saw that. If you go back like to the previous slide. One more. Wait, you go back. Yeah. See, where is it? See on the left-hand side underneath the biological assembly, it has global symmetry, asymmetric, global stoichiometry, heterotumer. MER. So I'm wondering if that meant you got A1B1, but it's actually a dimer. I'm wondering if that's why.
Right. It does seem like something that
Yeah.
should be, well, for me. What do I know?
But I don't know either.
I
Yeah.
don't know.
We'll ask
Okay.
Kevin.
So that's the answer to that. That there are in fact 21 in chain A and 30 in chain B. But
I
it's
just have
dangerous-
I
Go
just
ahead.
have a quick question, because when I was doing it, all I could find was chain A. I mean, all I could look at
Oh, so
was chain
that's-
A. I never found chain B.
Oh, it's a drop down menu right over here. I have it circled here. So there's a little drop down, and once you're in, hold on, let me go back. Once you're in, and what we should do is figure out a way to, like, in real time walk people through the PDB. We've never done that before. But so Judith, what I did was, by clicking on here where it says structure, right down here, that leads me to the next. Where am I? There we go. It leads me to here, and there's a drop down. There's a little arrow right here. So this is displaying the A, and then I clicked the drop down menu, and this is now just displaying the B.
Okay, I only found the 21. I never found the other.
Right.
I did find the dimer though, but I didn't find the
Oh, wow. Okay. So then, and I'm very proud of myself, because I think the last time I did this, we ran out of time and I didn't get this answer. The bonus question: how is the primary structure from 1 TRZ different from 4 INS? And so the answer here, this is from human insulin. 1 TRZ is human insulin, and the 4 INS is from the pigs. And what I knew to do, if I wanted to find out exactly the difference in amino acid sequences, I had to go to the FASTA sequence. Now, there might be another way of getting this answer, but I know what one at least led me to the right one. So on the main page, this display files, we're going to use this when we start inputting things into Phoenix and Coot. This is our PDB format file. I don't know if we use the MMCIF, but I know we use this. And you're going to need the FASTA sequence in order to create that ball and stick model of the protein that you're going to try to fit into that blue cloud. So I'm like, well, I know where I can get exactly what they are all in one spot. So there's the FASTA sequence. So I clicked on that for 1 TRZ and got this. So this is chain A's and chain B. So it's very short. There's only 21 letters here. Each letter represents a different amino acid. And then I did the same thing for the pig insulin. And this is the 2 chain A over here and chain B over here for, never mind. I'm sorry. This was the pig one. So scrofa and human is here. And going through it letter by letter, maybe there's an easier way of doing it, but I didn't find it. The very last one of chain B is an alanine for the pig and a threonine for the human. And so, yay me, I think I got the answer. I'm pretty sure that's it.
Well, I guess I was lost because I never found chain B, so I never saw a difference.
Did you find a different difference, though?
What I found was the assembly itself. There were 2 assemblies. For one of them, I think for the 4 INS, had assembly 1 and assembly 2, and it showed the connection between the 2 chains was different.
Okay.
And you could match the 2 different assemblies, but that was the only thing I found.
How about the kids? Anybody find anything better? Because you guys, I'm hoping, are better at this than us. All right. Well, that leads to that bonus. That's it. I did my job.
And you did a great job. Go ahead, Andrea. Go ahead.
I was gonna say, Kelly, that you can do this live if you just open the tab in your, Chrome and then share that tab. You can do this live.
Right. I'm sure. We've just never prepared. Like I guess we could. I know that there's a way to do it. We just never did that. We never said, well, today we're gonna walk them through the PDB, like in real time. And
Right.
I suppose we might want to think about doing that in the future because we've just never done it.
Mhm.
Okay. I'm going to stop presenting.
Right. Well, while you were doing that, I quickly jumped to the PDB. So here it is. One of the things I also wanted to mention, so here it is in real time. Here's the structure that you were just showing everybody. this is where we can get the papers from. A lot of these structures will have a published paper with them. And because we're talking about how do we get the information? How do we get the background on it? Well, this is how we get it. We click right here, and there's a publication that's called The Structure of Two Zinc Pig Insulin Crystals, and we can click on that there, and that allows us to go right to the paper. There's also an abstract that talks about it, and I was trying to read the abstract a bit to see what was going on with it, and it has chains A and C. So I think that's why you've got the sequence length there. But then I was also reading the paper, and it was talking about there were 6 insulin molecules in this particular structure they did in the unit cell. So I think that's the reason why they were able to. Oh, here it is right here. Can you guys see my screen if I highlight this? I don't know if you can see that.
Yep.
Yep. It says the positions and the contacts of the residues belong to the 2 A and B chains. And the details and arrangement in the 2 insulin molecules, 1 and 2, which are different. So that's why there are 102 residues. There were 2 in there. I was just trying to find that. But for everyone that's on right now, this is where we can get some of the papers from. So a lot of the proteins that we're looking at have, as Ms. Mackey said, there are cousins to them that are in the PDB. So we can go to the PDB, we can pull those articles if they're there, and that's where we can do our background research on the history of these proteins and what do they actually do, what's their function, what kind of diseases do they impart on people, what is the actual bacteria itself, where is it found, what's its role in the environment and all that. So we can get a lot of information from there as well. And then, of course, we have a standard Google search, which now can really give you lots of great papers from peer-reviewed articles. Many articles are available. Some that aren't, we can simply email the title to Sharon, and Sharon can usually find articles for us, and we can get those as well. So just because something isn't accessible to us through the school side of things doesn't mean that it's not accessible through B&L, so they can help us with that as well. So there's a lot that's in here, and it's a lot that Mrs. Mackey went over. But again, we're going to be using this as a tool so we can find the files that we need. We can find the background on the structure. We can find the history of it. And also just, those searches that she was talking about in the beginning are really important because we can immediately find out how many structures are in the PDB, how many structures of our actual protein from the bacteria that we're looking at are in there, and how many similar structures are in there from other ones that are similar to our bacteria. So it really does provide a tremendous amount of information that we can then use to help build our literature review, because ultimately the background information that we research on this is what's going to allow us to basically become the resident experts on these proteins while we communicate this information to the scientific community once we're all done. And that's what's going to help us communicate clearly and establish a really solid structure. Okay. Any other questions or comments from anyone in the group?
How long does it take to publish?
So to publish to the PDB, not very long. Once we have the structure, it's really about how long it takes to get the structure resolved to the point where it's publishable. And being publishable means we have the lowest resolution, the best resolution we have. It's complete, and it meets all the criteria for putting it to the PDB. And that usually is based upon Kevin taking a look at it and Vivian taking a look at it and saying, yep, okay, this is good to go. So basically, it's probably, I would say, 3 to 6 months once we say, okay, we're done with collecting data. Now we're going to resolve the data, and then we're going to process it and get that out to Kevin. So 3 months is really short. I'd say 6 months. I would say, we've probably started doing having the data in September, and then by I want to say, about 6 months later, like June or July, actually, we were able to deposit. And it depends. Each protein is different. Now, to actually publish the paper, that's another story. That takes sometimes a couple of years, because very often the writing process is long, and then there's the revision process. We submitted a paper, for example, to a journal. They gave it back to us, and they gave us suggestions. We went through the review process. Now we went through that, and now we're in the process of finalizing that and getting ready to put that out to a different type of publication. I hope that answers the question. Okay, we're good? All right. I think that's it, right? We're all set for today. Wednesday, we're going to learn a little bit about the synchrotron and what the synchrotron light source actually does in detail. I gave a brief introductory overview last week when we were talking about the reasons why we use X-rays. But on Wednesday, we're going to go a little bit deeper into that. And then on Friday, we're going to get together and kind of wrap things up. Sharon's going to explain a little bit more of the details of what goes into being a part of the collaboration and getting access to the lab and the important things that need to be done. And then we're going to talk about our plans as a group and moving forward, the workflow that we have planned out. So we're really getting to the point now where we're going to start moving ahead now. We're going to start diving into these proteins. I'm getting excited about that. Should be good stuff. So that's it. So students, you can log off. Have a great day. We'll see everybody on Wednesday at 10 a.m.